Thanks a lot, Lisa, and welcome everybody to a fun evening of parasites. I'm going to talk about global parasite biodiversity, which is basically the thing that really informs our book, which really informed the development of our book. And so we call it the inside story because it sounds kind of like inside, endoparasite type thing, I guess. So I'm going to start off, just go full speed here. Biodiversity. Biodiversity is, the definition is the diversity of life in a defined area, from genes to genomes to organisms, populations, communities, ecosystems, biomes, and of course, just on our Earth. In this picture, we have a really diverse group of things. We have a tuco, rodent of the family Tenomyidae up there to the left, and then we have a strigiid cercaria in the middle, and a frog there, a tapeworm, a forest, and some chromosomes. So basically, we have a lot of biodiversity if we delved into what kind of parasites live in these things. And of course, there are two parasites. This is one parasite here. I'll get up my laser pointer. This is the strigiid parasite that comes out of a snail, that then tries to go into probably a bird or a turtle. This is the head end of a tapeworm. So let's keep going. So today, the goals are to give an idea of what it takes to do a parasite vertebrate survey anywhere in the world. What really does it take to do that kind of work? We'll discuss problems of emerging parasitic zoonoses. A zoonosis is a parasite that or other pathogen that lives in or on wild animals and can be transferred to humans. And then I'll summarize some of our work on cestodes in the Nearctic region, which is mostly North America. Also the Neotropics, which is South America, and then Central America as the southern or the northern part of the Neotropics. We'll also talk a little bit about what we have found in the Palearctic, both Europe and mostly Asia. Then we'll talk about some details of one of the most pathogenic species of tapeworms, of cestodes that exist, and that's called Echinococcus. We'll talk about that a little further on in the talk. Then there's more goals. So we really want to emphasize some of the depth of information that we store in our biological collections, like the Manter Lab. Of course, the British Museum is much deeper than what we have here, but we have quite a lot. I also want to show how biologists are using specimens in the collections and in associated collections in new ways, and why we do this and how we actually produce a synthesis, time-consuming work, dangerous field work. Sometimes the material won't be used for many years. Why are we doing this work ourselves? So then the last part, which I'll start with first here, is called DAMA, a protocol that biologists can use to coordinate efforts in biodiversity inventory, parasite pathogen identification, and response. So basically, the DAMA protocol is something that we developed to basically make a protocol that aims at dealing with emerging parasitic diseases or even virus diseases, as well as biodiversity loss. So DAMA means D-A-M-A, document, which means surveys, what is where, what kind of parasites live where they are. Assessment, discoveries of the different kinds of patterns that we might see as we're assessing what the documentation phase of our work would be. And then monitoring, which is another part, is watch what's going on. So is something new coming in? Are we seeing in different areas of the Earth, are we seeing ecological changes? And if ecological changes are occurring in one area, maybe we need to go to another area that we're also monitoring and see if it's happening there. Then, of course, the monitoring part means that we need to go back every once in a while to see what might be there, what might be changing. So looping back to original areas to check status of biodiversity. And then we need to then act. The point is, if we do these things, document, assess, monitor, but what about the action part? And as far as I can tell from all of our work so far, that the action part, making public policy changes, is probably the most difficult of this whole thing. You know, this stuff is hard in just by itself to document parasites and hosts over space to assess them, to monitor them. But then the action part is, to me, difficult. The action part here can be to prevent an outbreak of zoonosis that might be happening. For instance, rabies. We might see a rabies outbreak somewhere, or a echinococcus outbreak. I'll talk about that a little bit later, which is a hydatid disease. The idea for DAMA is to provide real-time reports on ecosystem health. That's something that could happen if it was implemented properly. And one of the things that it could do for the action would be to prevent ecosystem collapse with advanced warning. So we might see things changing rapidly in one area, and we might be able to do something about it. I'm not sure what that would be, but at least we can say that it might happen. We can also stop an outbreak that's currently underway, and I'll provide an example of that in Hungary. All right, so let's start off with the documentation part. And this is basically broad-scale biological diversity surveys and inventories. And this is some of the things that we've done since 1984, when I first started going into the field. The specimens that we bring back are correlated voucher specimens that are deposited in museums. We get parasites from a mammal; those go in. The parasites are in one museum, the mammals might be in a different one, or they might be in the same one, but we can link those easily over the internet with databases, correlating georeference collection localities, mammals, parasites, field notes, images, and all kinds of different things. And those are shared over the internet with anybody who wants them, basically, because we're a public institution, we are able to share these things. And we share them via GBIF, which is the Global Biological Information Facility, Arctos, BirdNet, the Biodiversity Information System for Europe, which is BISE, Encyclopedia of Life, and others. So those things are coming. We put our data into our collections, their database, and then they're available for people to use and understand. Additional documentation. Basically, I think I just had gone through that. The assessment part is the the part that is a little bit more difficult than the documentation part, because we have to actually see what's there. And so we want with the assessment is to get species-specific information for the things that we're collecting in the field, including ecology, behavior, biogeography, epidemiology, and more. So all those things, the data are coming in as we're doing our collections, and the assessments are going on. And also we put it into the context of phylogeny. And the context of phylogeny is important. And we kind of base that on what Manter called Parascript in 1966, in which parasites tell not only their own story of ecology and evolution, but each parasite tells the story of its host and geographic distributions. And I added on to his little summary there, and the evolution of complex ecosystems. So Manter in 1966 was prescient. He was thinking into the future about how to use phylogenetic systematics and ecology to understand problems in the biosphere. And the lab is named after him. He's an amazing person. So more assessment. Part of the assessment involves rotating around what we now call the Stockholm paradigm, which was developed by Dan Brooks and colleagues. And this is a central part to the assessment of the DAMA protocol. Actually, the DAMA protocol is subsumed under the Stockholm paradigm. And this is based on ecological fitting, which is basically host parasite transfers, something called the oscillation hypothesis, which is developed by Jens and Nyland, and then something called sloppy fitness space, in which parasites are occupying what they can, and they can also jump into new areas when they want to, or if they have the ability, or if they have the opportunity to. And that was put out by Salagosta and Clemens in 2008. Then, of course, taxon pulse, in which we see increases and decreases of species diversity over time by a guy named Terry Irwin, who worked on beetles in the tropical rainforest. It's been developed further by Hollis in 2005. And so basically, the Stockholm paradigm is something that's been recently developed to explain the dynamics of origin and maintenance of diversity of host parasite systems. And I include parasites as viruses are part of the parasite idea here. So I'm going to just give some little examples here using some of my favorite animals. We can see here, there's under H2, it says that's a tuco. That's an animal called Tenomys lewisi that lives in southern Bolivia in the department of what's called Tarija. And there's another animal there that's called a tuco. It's called Tenomys conoveri, and it lives down near Paraguay. You can see there's a little handheld pliers below that tuco. It kind of was there for a size measure. But this is just a hypothetical example to show how ecological fitting or host switching works. You can see the phylogeny on the left side here. I'll show it. This is the phylogeny. It's basically a host and parasite phylogeny, the host 1, host 2, host 3, etc. The parasites are the light blue lines. If we had marked them, they would each parasite species would be different. But this basically just shows what ecological fitting is, and it shows that a parasite that's in host number 2 makes the jump to host number 6. And even though host number 2 and host number 6 are far away from each other on the phylogenetic tree, the common ancestor was here, and there were several common ancestors before host number 6. It shows that there was a transfer of parasite through space. So this happens many times in nature, because animals that are evolving and moving through geographic space come into contact with each other, and they can pick up each other's parasites. So one of the points about all this is that co-speciation and not necessarily coevolution is what's shown here. Coevolution is what was termed by Dan Jansen was reciprocal evolutionary change. And that means the parasite affects the host affects the parasite, and so there's evolutionary change with co-evolving species. But it's very, very difficult to show coevolution when you're doing field-based collection work, because we have to show reciprocal interactions, and it's very difficult to show that with the kinds of data that we've been collecting. I think experimentally it would be more appropriate for those kinds of things. So the monitoring part, so here we are assessment. Now we'll switch into monitoring. We'll reassess, loop back, and reevaluate previous surveys. So we're monitoring what's happening. We'll see what these data are used, what data can show to detect changes. Are there species missing from our species inventories? What is new? Are there new species moving in? Are things disappearing? Are there new transmission dynamics that are going on? And maybe there's new host records that are showing up. And so all these things are the monitoring part of the DAMA protocol. Another part of the DAMA protocol is to continue doing basic inventories, which is basically a continuing process, compare findings among localities across timescales. And that's really an important part of this. We can't just go into a geographic area at one time, collect, and then never go back, because we don't know what the changes might be. So if we find changes, if we see species moving in, if we see certain things happening, changes are detected, that will alert us that something odd is going on or something new is happening. So if we see something in one area, that might alert us to look carefully in other areas to see if the same thing is occurring. So that whole thing should enable us to anticipate similar changes in other areas if we see changes in one area. So those are the ideas for the monitoring part of the DAMA protocol. As I said at the beginning, the action part is probably the most difficult, because that is the political part where we have to get people to agree to do something about changes in the ecosystem or changes in biodiversity that are occurring. So in this part, I say the action part is to develop policies that monitor and use parasites as indicators of biodiversity, climate change, and emerging diseases. And these policies must be proactive. So they must use data accumulated to make preemptive decisions. If things are starting to happen, we have to be able to make a decision. The action part also has to be anticipatory. So we have to be able to use these data to anticipate outbreaks, including emerging zoonoses, like SARS, for instance. So the COVID-19 pandemic is something that jumped from zoonosis, from a zoonotic origin into humans. And so I'm not a virologist, but I'm a parasitologist, but we can speak about the same things. Also, the actions might should be large scale. So we sample for parasites over wide areas. Okay, so the action part here is to implement policy to enable proactive discovery of parasites and viruses that will or may jump hosts. And again, this is to just show how parasites jump from one host group to another. And I think that it would be interesting if we actually have enough data from some of these areas of intense survey that we could actually produce forecasts of probabilities of ecological fitting. What I mean by that is we can actually have forecasts of pathogens erupting in an area. If we see a lot of mosquitoes coming into an area that haven't been there before, and those mosquitoes are good hosts for like Zika virus or hantavirus is coming up in some of the mice that are in the areas. If we have more mice in an area that are normally there because of a wet year, then we could put out alerts saying this might be happening. Lyme disease, of course, is caused by Borrelia. It's a bacteria that is transmitted by ticks. So we have all kinds of things that we could be monitoring and make proactive, predictive decisions on. Plant pathogens, of course, are another thing that we should not ignore, because plant pathogens are vectored by pests of crop plants like leaf hoppers and various things like that can transfer viruses from plant to plant. So all these things can be done. It's just a matter of doing it. So, so far there's been one really well documented implementation of Dunne, and this was done by Gabor Racz at the Institute of Evolution, Center for Ecological Research, and Center for Eco-Epidemiology and National Health Security in Hungary. And he started working on some islands in the area of the Danube River in Budapest, and people were coming down with Lyme disease there. So he went in and studied the ticks that were transferring the Lyme disease to people. He determined what the abundance of ticks were and where the ticks were most common, and he came up with an idea of how to solve that problem. So he documented the problem of the Lyme disease in humans in Budapest, and also in mammals that are living there, other mammals, I mean. And they assessed the problem. They looked at ticks, various prevalences of infection with Lyme disease, spirocate, Borrelia, over a geographic space in these areas where people went to picnic on the islands. And so then he monitored what was going on. He checked the cycles and the hosts, and they actually discovered more than one species of Borrelia there. I think it was like 5. And then he proposed action, and the government implemented the action, and it was an easy solution. They built public toilets. They kept people from going out in the bushes and picking up ticks. So that was the solution to decreasing the number of people becoming infected with Lyme disease in those public park areas in Budapest. And that was, I think, a resounding success of the Dunne protocol. So we're going to leave here. We're going to go into the field quickly. In this case, Western Mongolia. So this is one of our field camps in the western part of Mongolia. You can see in this image, this is our Giersche. This is where we have our laboratory set up in the Giersche, and we have another, I guess maybe the laboratory was set up in this tent this year, and our Giersche was set up here for our kitchen work. Subsequent years, we didn't use the tent because it just couldn't hold up under the wind there. So in the field, we collect data. This is an example of a data point. This is a hedgehog, and we got it at this collection locality, which shows up on this map. Gabor does the work to find out what's there. This is a picture of Gabor working in the lab inside the Giersche, and we euthanize the animal. We collect the parasites. We make a study skin from the animal. We collect all the parasites, either go into liquid nitrogen or other vials of formalin or alcohol. And so this is me working in the field with our microscopes in the field there too. We have specimens that are going into different vials to bring back into the lab so they can be used for the future. In this case, this shows when we were doing our work, we were collecting birds, mammals, and reptiles and amphibians. And so this is the tray of birds over on the left here. This is part of the sample that we collected in Mongolia. And then over here shows some of the mammals that we collected. At the time of collection, we collect some parts of the parasites in various types of liquids like formalin to preserve for morphology, or we might use what's called RNA later, which preserves the DNA at room temperature or at refrigerator temperatures. We also save some of the specimens in liquid nitrogen stored at minus 80 degrees centigrade. It's actually colder when we put it in the tank. This is one of our tanks. And this is one of our ultra cold freezers, which we preserve all the specimens in, that we can then use DNA sampling or DNA sequence analysis to find out how these things are related to other parasites or other mammals that have been collected over the face of the earth. So we have a lot of data that are coming in. In this case, these are nematodes in these vials, and you can see we have labels in the vial. This looks like maybe it's some tissue or perhaps a tapeworm. So we have all these vials with all these different samples in them, and so those all have numbers. The specimens are then available in our collection once we've cataloged them, and this is what the collection looks like. So we have tray upon tray upon tray of vials. These vials are big ones up here. All these are the same size. And so each vial has a barcode number on it, so we can use a little scanner to scan the vial to know what's in it. We can't keep our collection like vertebrate collections. Vertebrate people keep their collections with all this families of birds in one cabinet, all the families and genera of rodents that are similar in another cabinet. But these things are so small, we can't just keep them like that. So we have to keep them in vials, and they're all numbered. And so all these are correlated on our database. If our database ever went away, we wouldn't be in very good shape, but we have massive numbers of backups with this. So this is what we call the vial storage facility. And this is just one cabinet out of about 20 of them that we have. They're different sizes too. So what do we do? So this is a picture of one of my former graduate students, Mary L. Campbell, and she's looking at tapeworms that we collected in Bolivia at this point. So we look at the specimens that are on slides, and she's looking at a slide here. And then we look at the specimen. This is actually a specimen that we analyzed using a microscope. We can see the scolex. This is the head end of the tapeworm. This part right here is right inside this. This is called the apical organ. This is the serous of the tapeworm. That's the intermittent organ of the tapeworm, so it can mate with another segment. This is an egg. We see hooks on the egg. This allows the egg to tear through the hemosyl of an insect once the insect or acarine mite eats it. And this is what the tapeworm looks like. If we expand up one of these sections that's in the mature area, we would see these parts and pieces. And so these are the testes, this is ovary, this is the vitelline gland. And there will be an exam afterwards, so make sure you memorize those. Just kidding. This is one segment that has all the eggs inside. So these segments are repeating segments. They're metameric segments that go, they keep growing at the neck region. This is the fastest growing part of a tapeworm. Anyway, I could go on and on about this, but we have a lot more to talk about. So here's me. The specimens in those vials eventually end up on slides, and I'm looking at a tapeworm. I think that might be the same one that I had made the drawing of there. This is one in a box. So all these cabinets are full of boxes, and each box is full of slides. So we have literally hundreds of thousands of slides with specimens on them. Our microscope, donated to us by Zeiss. We have this beautiful scope, and these are examples of some of the protozoans that we have in our collections that we can also study. So the question is, why collect natural history data? A lot of people say, well, what good is all this stuff? Well, one of the things that's happening is that we as humans are building and wiping out the natural world basically as fast as we can do it. Unless we collect the data now, there really won't be anything left for our descendants to look at or know about or to really wonder about. And so we have to do the collections fairly soon, because if we don't do it, things are going to be so changed, we won't be able to get information that represents natural systems. Basically, if we don't work to describe and conserve biodiversity now, our descendants will be very upset. So they'll wonder why we didn't do something when we had the opportunity to do it. So the question is, why vertebrates and their parasites? Why not just go collect the vertebrates? Why deal with the heart, with the complexity of looking at their parasites also? And so this is one area where we can really tell you what's happening with parasite host biodiversity. So what happens is many of the parasites can be indicators of biodiversity. If we find many parasites with complex life histories, that means that the parasite eggs come out of the host, they go into something like a beetle or a mite, and then the beetle or mite, then they develop inside the beetle or mite. And this, with lots of parasites with complex life histories, indicates that this might be an area of high biological diversity. So tapeworms all use intermediate hosts, all those that we know of, except for a couple of species. We can also infer, based on phylogenetic analysis, that parasite species occurrence information over geographic space can provide information on deep geological time, which I think is pretty interesting. I'm going to show this fairly quickly. This is kind of how we would imagine if we didn't know anything about, well, there's a mammal there, but we identify the mammal. We know it's a tuco, it's a Tenomys lewisi. We don't know anything else about how these are related to anything else. So we have no patterns. We have no prediction. We can't make any idea. We don't have an idea of what's going on. So if we go through time, as we collect parasites, we identify them, we can get a lot of information. So at the end of what we're trying to do is we increase our knowledge. We're looking at patterns and processes. We're looking at patterns and we're coming up with processes that make those patterns. And that gives us predictive capabilities. So we can actually make predictions about what might be happening. All of these data that go into museums, ectoparasites, helminths, protistans, viruses, all those go into the museum and the databases, and they can be then shared over the internet, auto-relational distributed GIS databases. That's the dream, is to have that. It doesn't occur yet, but we hope it will. And so those will then be used by researchers in the field. So I'm going to give some examples of research that informed our book. The work that's done in the lab has been done over many years, starting off when I first started to become a biologist. I'm going to talk about some example studies of biodiversity and systematics. And one of the first things I'll talk about is using parasites as probes for biodiversity. Then I'll talk a little bit about Echinococcus, which is a really severe human pathogen. And at the end, Gabor will mention some research that's been done on using parasites as probes for human migration. So the reason we do all this is because of this. This is the only place that we know life exists. There's speculation that it might be somewhere else, but the probability of us finding that is going to be pretty low. So here we are, right here, where all this stuff that's happening is right here. And this is a picture from the moon of one of the Apollo missions. So talk about tucos now, Scott. Here we are. So we're going to now talk about tucos briefly. And this is one of the rodents. Tucos are one of the rodents that I first started working on in South America when I started doing my dissertation work in Bolivia. So here's a picture of South America from Google Earth, and Bolivia is shown there, pointed at an arrow. You can see it's one of the only landlocked. Well, Paraguay and Bolivia are landlocked countries in South America. They don't have access to the coast. One of the things is, what do we accomplish with biodiversity surveys? And just looking at only the tucos, only the Tenomys, these are the animals that are called Tenomys. This was what we knew about them in 1984 when we first started working there. There's Tenomys boliviensis. Let's see here, laser pointer. Boliviensis occurred in this area generally. Leucodon occurred in this general area. Opymus on the Altiplano here. Lewisi down in this area of Chuquisaca and in Torija. Tenomys frater. So we generally had an idea what was there. And in Brazil, we knew that there were tucos there, but we didn't know that much about them. After our work that finished about in 2000, intense work, we're still working there with colleagues, but our intense work finished about then. This is what we knew after our biodiversity survey. So we know where Leucodon occurs. We know where Opymus occurs. Tenomys lesi, which is a new species we described from Bolivia, occurs only in this part of Cochabamba that we found so far. Very restricted distribution. Lewisii, Tenomys lewisii is very small place and geographic extent here. Tenomys conoverii also occurs down into Paraguay, but my drawing doesn't show that, but it occurs here. We didn't work in any other country while we were there, just in Bolivia. So we see that we have a lot more data. These are the animals. This is Leucodon on the Altiplano. This is Tenomys lewisii. This guy's called Tenomys. This is Tenomys leucodon, Tenomys conoverii, a really big, very interesting tuco. And this one is called Tenomys frater. And Tenomys frater occurs right in this area of Bolivia. So we'll continue here. What are tucos anyway? So this is an example of a tuco. It's Tenomys lewisii. Lewis is tuco. And this is some examples of Tenomys conoverii, some different shots. This one has really shaggy fur. They have great big teeth for burrowing and cutting plants. They have, the reason they're called tenomys is because they have combs on their feet that allow them to kick the soil out of their burrow backwards. So that's why they're called tenomys. Teno means comb, mes means foot, tenomys. So why parasites in the vertebrates? Well, parasites as indicators of biodiversity is the important part of what we do, trying to understand the parasites with complex histories. Tapeworms almost all use intermediate hosts. Of all the tapeworms that we know, there's only a couple that develop straight through and are able to develop and produce eggs without going through an intermediate host. Parasite data can provide deep geological time information. I've said that before, but this is an important part. And one of the things that's important to know is that not much is known of the ecological, phylogenetic, or genetic diversity of most parasites of wild birds or mammals. It's really unknown out there. So another part of DAMA is this. How can we use data from past collections worldwide to help solve environmental problems today? And that's the assessment part. And I'm going to go ahead and talk now about using parasites as probes for biodiversity. And the point I wanted to point here is, are the areas of our study still connected in function, even though they're disconnected in time and space? And so museum collections are important here for monitoring this. And so I'm going to talk about that now. So parasites as probes for biodiversity, and this is in the sense of Gardner and Campbell, 1986. So this is kind of delve into parasites with complex life cycles. The reason I think that they're important indicators of areas of high biodiversity is because a host like this small marsupial produces eggs when it's infected with tapeworms. The eggs come out in the feces, and we don't know what the intermediate host is, but we know it has to be some type of arthropod. So it's maybe an orabatid mite, which is a mite that lives in the soil, or it might be springtails or collembola. Collembola are little, very, very small soil organisms, and they've been shown to be an intermediate host for tapeworms in the Palearctic region. So then the intermediate host is ingested probably by either because these guys are insectivorous or they're eaten accidentally. So that's how the cycle goes. So then what we decided to do was we did a phylogenetic analysis of the host and the parasites combined with integrating historical ecology, ecological survey data, biogeographic analysis, and we even looked at continental drift. So here's this is the interesting part. Continental margins are subducting. The Pacific plate is subducting under the South American plate. The Andes began to lift up about 80 million years ago to the present. In here, we have a lot of variability in the topographic landscape. So in this area, there's what's called a central Andean backthrust belt of the mountains here. And so as the plates are subducting here, this plate is going this way, and these causing these mountains to lift up, and it's making these folded ridges of mountains out into the lowlands of Brazil. And so we look at a cross section. Imagine we did a cross section of South America, and we're looking towards North America to the north. This is what it would look like. Here's the ocean in Chile. And then we go almost straight up to the tops of the mountains at 6,000 meters. And the altiplano is about 3,500 to 4,000 meters, which is the flat part up there. And then we drop off towards Brazil through these areas that are forming these big folded crustal zones. So the area where we were working is called the Yungas, which is this area here. This is what it looks like. If you look at a road I'm on the road, and that truck has to come all the way up to where I am after a circuitous route. So it's pretty steep down there, pretty dangerous driving. This is what Bolivia looked like when we look at only the collections localities of the marsupials from the area where we were working. So we collected all of Bolivia for our survey. We were working with the Bolivian museums down there. We were working together with all these students. We always had about 12 students with us each time. But all this work, we found tapeworms here, here, and here. We didn't find them on the altiplano. We didn't find them down here in the lowlands. We found them just in these three places. And these are in the Yungas, the front range of the Andes. As I said, yes. So in those areas, we removed the rest of the collections. We can see that they're right here. And they go from about 1,200 meters or less here up to about 2,200 meters at this point. As we get closer to the equator, we move up in altitude to have the same kind of environment. So I'm going to have to move relatively quickly, so I see that we have a lot to cover. So I made a phylogeny. Mary Ellen and I made a phylogeny of the Australian and neotropical things that we call Linstoia. And there's still some debate about how these things are classified, but we'll call them Linstoia for now. And it turns out that all of the species in South America had a common ancestor here, and all those that occur in Australia and New Guinea had a common ancestor down here. And so there's been some diversification, but there's definitely a group of species that occur in Bolivia along this area, and they are separated from those occur in Australia. So what's going on? So this is what I think is going on, is that there's a mountain range. 180 million years shows here, but about 60 million years ago, when the the meteorite hit about 80 million years ago here and took out the dinosaurs, the continents had started to move apart further. But this is what it looked like. Originally, these are kind of some mountainous areas that are shown here. But about 80 million years ago to 40 million years ago, they started to separate. So you can still see this mountainous region. So in this area, there was still habitable. So the marsupials were still distributed well all across this area. And let's see if I can make this go. Yeah, so the life cycle is occurring in this area, not 180 million years ago, but about 60 million years ago when these continents were first started to come apart. So that's the Jurassic. So later on, 40 million years ago, when South America was an isolated continent and everything had started to split, the life cycles were still still going. So the interesting thing about this is that I feel that this is an indication of deep ecological time where we're seeing life cycles still going in these two areas, Eastern Australia and Western Bolivia. And the cool thing is that really what we're looking at when we're looking at these kinds of tapeworms, we're looking at soil faunal diversity. Probably what we're really seeing is the diversity of the soil fauna, because that's showing what is necessary to be there for these tapeworms to still be cycling. So also the discovery of cestodes in some, but not all of the marsupials surveyed indicates that additional data should be collected in the Yungas to find out what else is going on in there. If we go through the whole part of Bolivia, we only see that those parasites are in the Yungas, in those mountainous regions. Therefore, it gave us the idea that they're probably related to old ecosystem in Australia that's still cycling. So this is the paper that we published back in 1992, Parasites as Probes for Biodiversity. This is probably like the fifth time that the word biodiversity was used in any scientific paper. So let's go on. I'm going to talk a little bit now about zoonotic diseases. Zoonotic diseases is an agent of disease that circulates in wild animals, transmitted from human to humans by some vector. Vector can be air, it can be water, or it could be a bite from a rabid dog, for instance. So a reservoir of a zoonosis is a wild animal population. That's why it's called zoonosis. As I said, a vector is anything that can be transmitting. So during our Bolivian biodiversity survey, we found we worked all over Bolivia collecting these little marks here. These were where we found pacas, agouti pacas. We only found this parasite in one spot in Bolivia, but we now know that it's distributed much more widely. And so to identify it, this is before we had access to DNA easily, but these are the hooks on the rostellum of the larval tapeworm. So we were able to identify it fairly quickly. So now I'm going to talk a little bit about the problem of Echinococcus. And this says, move to the other side of the world, Scott. So we're now going to move all the way to Mongolia. So here we are. So we're going to go from where we are right now, and we were all the way down here in Bolivia, but now we're going to go to Mongolia, and we're going to just go through some of the data that we found in Mongolia while we were working there. So we did a 4-year project in Mongolia to do a biological survey of the mammals, birds, and herps, which are reptiles and amphibians of Mongolia. We went all over the place, and basically the western part of Mongolia. We didn't get up here yet, but this was done at a later time by 2012. 2012, we went all over the place, but we found the parasite that I'm going to talk about only in one locality, even after all that work. This is the parasite. It's called Echinococcus multilocularis. It's easy to identify. It has hooks on the scolex, on the rostellum. It's got 4 suckers, 2 here on this side, and if you go over the other side, there's 2 more. It's these animals, when they're adults, are full of eggs, and these are the things that cause the problem. Because if you are a vole and you eat some feces from a dog or a fox or anything else like a cat, but we're going to talk specifically about Echinococcus multilocularis here in Mongolia. If you're a vole and you eat some of these feces and the dog was infected, then the vole becomes infected, and then the larvae begin to develop in the liver or other tissues of the animal. I'm not going to talk about the species that occur in moose or other ruminants at this point, and I'm only going to talk about here multilocularis with just a little bit of information on the South American species. Well, multilocularis occurs throughout Mongolia, all the way through Russia, all the way through northern Asia, all the way through northern Asia into North America. So it's a very common parasite that occurs in wolves, foxes, and voles there. So Mongolia, this is kind of where we went. We studied everything from this side over. And this shows what the habitats look like. This is mostly steppe and desert steppe region where we were doing our work, because not much had been done down there before. So we had a grant proposal that was funded to do that work. We found the Echinococcus vole. We only found one vole of all that we had collected over all those years here at a place called Har Us Nur on near Jargalant Mountain. And the habitat looked like this. So this is up on a hill where we were away from the mosquitoes at our camp. And down here is where we had our trap set out down here next to the lake, where the mosquitoes were so intense that we would have been exsanguinated in just a little while if we'd gone down there. This is what the habitat looked like where the tapeworm was found in the vole. This is one of the people who were living there. They were just getting ready to leave. They left the next day because the mosquitoes were so intense by that time. They moved to their summer quarters. The vole is Microtus, called Microtus limnophilus. They occur in all over that part of Asia. And we open the vole up, and there is its liver, and its liver was basically non-existent. You can see here, you see all these cysts in the liver. So we went further, we took the liver out along with the intestines, and you can see the only thing that's left of the liver of this animal is a little tiny piece right here, and maybe a small one here. About 90% of the liver has been transformed into tapeworm cysts. And inside the tapeworm cysts are these things called protoscolices. There's one here. When a vole gets eaten by a fox or a wolf, this protoscolex then turns into a tapeworm. And so there's thousands of them inside. So when we look at this protoscolex, we can see that there's hooks inside them. So when we take one of these protoscolices out, put it in lactophenol, which is a clearing reagent, and then squish it on a microscope slide, we can actually see the hooks to measure them and identify them. We also used DNA, COX-1 and ITS-1 and 2 to identify these things. And so we made a positive identification that multilocularis did occur in Mongolia. That was the first record from a vole there. Okay, one of the things that's interesting in North America, we don't know really much at all about what's happening. There's either an advancing front of multilocularis that originated up here in North Dakota. It's only been found a few times in Nebraska. We reported it from Park in downtown Maryland, Baltimore, Maryland, in fact, and we also found a record in New Mexico. One of the mice that were sampled in New Mexico turned out to be infected with multilocularis, and that had never been reported before either. It was only thought to be up here, but now we know it's all the way to here. So the question is it advancing south as the climate warms, or is it going to retreat as the climate warms? So this is why we need to use ecological niche modeling based on data from parasite collections to really understand what's happening. So that's another area of research that we've been working. Just quickly for everybody's, so everybody will know, there's several species of Echinococcus granulosus, canadensis, multilocularis, Schweitzer, Fogeli, Oligarthrus, and Felidus. These species occur in different places, and I'll just quickly go through them. We don't have much time. Granulosus is whole Arctic. It's moved around. Its wild form goes cycling through moose and wolf. Canadensis also occurs in ungulates and canids. Multilocularis occurs in the whole Arctic region, North America and Asia and Europe. Schweitzer occurs in Central Asia, Oligarthrus in South America, Fogeli in South America, and Felidus has been reported from lions in Africa, but we don't know what the host is, and it's never been reported from people there yet. So those are some unknowns about that one. The approximate distribution of these kind of shows here. This is an older map, but it shows Echinococcus in the central part of North America. It shows the different species in South America in these little blue squiggly lines. And in Africa, we see that Felidus occurs here. And then these dark areas are hyperendemic areas where all the dogs are infected or the wolves are infected. So now we're going to talk about, well, I think I can do this. We have just a minute. So the question that I had about this Echinococcus question is there a retreating or advancing front of infected voles and foxes and coyotes in the central Great Plains of North America? So we don't know. And so we have applied for funding to do the work to find out what's there, but we've not been successful yet. So we really need to know what's happening, because once these things move into areas where people are, different things can change. For instance, in Switzerland, the government decided to do a vaccination program for the foxes in Switzerland and decrease the number of infected foxes with rabies. So they dropped rabies vaccines from the air, and the foxes then all became they became immune to rabies. And at the same time, then their numbers started to increase. And as their numbers increased, then the foxes moved more closely to where people were living. And the foxes were infected with Echinococcus multilocularis, and people's dogs were getting infected by eating voles that were being infected by the wolves or by the foxes there. And so then people started coming down with echinococcus. So one consequence, an unintended consequence of rabies vaccine was increased numbers of people infected with echinococcus, which is a fatal disease unless it's treated. So let's see, I'm going the right way. No, I'm not. Okay, so now we're going to switch over, and Gabor is going to take over and talk a little bit about probes for using parasites as probes for human migration. So take it away, Gabor. I'll mute myself.
Hi, everyone. This will be just a few minutes. And on the next slide, so when I got to Nebraska, there were a paper published in Trends in Parasitology. It was published in 2008, and Scott was a co-author on this paper. Carr Reinhardt, also a University of Nebraska professor. And I would recommend to read this paper. Basically, this paper brings up the idea that if you are looking for fossil findings in North and South America, mummies and fossilized feces, the parasites that they were finding in these, the parasitic remains that were found in these fossil remnants indicate a different kind of migration route for humans to North America. On the next slides, usually what they are saying that humans reached North America and then South America through the Beringian Bridge. This assumes a kind of Arctic migration. But if you look at the parasites, namely hookworms, whipworms, and a third one, which is called threadworms, Strongyloides stercoralis, it argues for a different kind of migration route. Some of these parasites will not survive in Arctic environments, and they indicate either a coastal northern coastal route of very fast migration to North America, that's line B, or the Strongyloides stercolaris, which is a tropical human parasite, only occur in tropical and subtropical areas, that might indicate a trans-Pacific migration route for humans. And on the next slides, a few years, it missed, but yeah, this is the one that a few years ago they started to publish some papers on stone tool findings in North America, and they analyzed these arrowheads. And the conclusion of these 2019 and 2022 papers maybe was that they are most similar to other findings in Japan. So it would, and they were kind of arguing a coastal migration route for these early humans in North America. And based on the parasite findings, this is not surprising. And we are expecting that there will be additional findings that might even confirm a trans-Pacific migration route for humans. Back to you, Scott.
Thanks, Gabor. That was a well-stated summary of that topic. And so I think what we'll do now is we'll just basically give a summary of the whole thing. What I wanted to do today was define in general the concept of biological diversity. So I think I did that, and you can see that diversity is really intense when it comes to looking at parasites. We didn't go through all the different types of parasites you can find in one host, but it's pretty amazing. We also defined and explained what the Dunne protocol is, and we showed the importance of broad geographic sampling over geographic space to document biodiversity. Because if we had done that work in Bolivia, where we only collected marsupials from a couple of areas, we would never have known that only in the Yungas do the tapeworms occur. We also used parasites, showed the use of parasites to document and indicate deep time ecological connections with the species of Linstoia and the marsupials and the monotremes of Australia and the marsupials in South America. So I think that's pretty interesting that deep time ecology can be pulled out based on tapeworms. we also summarized a little bit of information on the pathogenic tapeworm Echinococcus, which I think is a really interesting one because we're right in Nebraska, right where it might be now. And we, as I said, we took a deep dive into mammal parasite diversity, and we touched on the use of parasites to understand prehistoric human migrations. And I think that that last part was, to me, pretty interesting, that we can actually use parasites to understand distribution of people in prehistoric times. So that's all I have right now. The reason we do all this work is because of this again. This is why we do the work, is because this is the only place that we've got. We need to know what's here. We have to save what's here. We have to work with it. So thanks everybody for being here. And I'll turn it over to our moderators.